Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Eurofins Pharma Discovery Services, with their partner lab, Pharmacology Discovery Services, I'd like to welcome you to Profiling an Oncology Drug with In Vitro Assays and In Vivo Models to better understand its potential clinical applications. My name is Elizabeth Lamb, and I'm the host and the moderator for today's event. Now I'd like to introduce our presenter for today. He is Dr. Alastair King, PhD, OncoPanel Cell-Based Assays Group Leader for Eurofins Pharma Discovery Services. Welcome, Alastair. The presenter ball is yours.
Well, thank you very much, Elizabeth, and welcome, everybody. It's a pleasure to be able to present to you today. As Elizabeth mentioned, I'm going to outline approaches where one can profile an oncology drug using a case study with a variety of in vitro assays and in vivo models, wherein the information gathered from those models allows a better understanding of potential clinical applications. So as we go, but before we go forward, I just want to give a quick overview of what we do here at Eurofins Pharma Discovery Services. This just gives a snapshot of some of the legacy companies that that became part of Eurofins. We have a wide diversity of backgrounds, and several of the organizations have been viewed as gold standards in the pharmaceutical industry, in particular the Sorafenib group in France, are well known for their safety pharmacology approaches, and the Upstate group that was part of EMD Millipore's discovery and development solutions have been considered a gold standard in the kinase world for many years. More recently, many of you may have heard, we acquired Pharma, which is a medicinal chemistry company in Spain, and most recently DiscoverX, which brings a whole new wealth of different assays to our portfolio. So with that, then I'd like to just give you a quick overview of the agenda for today. We're going to talk about kinases in drug discovery, and we're going to use a case study to show how one can put together different pieces of information from different assays and different models to better understand a particular kinase drug. So within that context, I'm going to introduce a therapeutic need, and we're going to use the drug Sorafenib as a case study for this. I'm going to show how different biochemical profiling of both Sorafenib and a couple of other compounds can actually provide information that can help guide interpretation as to the drug's profile and abilities. I'm also going to take a look at cell-based profiling that can provide a different depth of information around the drug and can also assist with understanding its profile from a biological sense. We're going to take a quick look at genomic predictive biomarkers and how those can be identified that, in addition to potentially streamlining and guiding clinical trials, can also help understand and explain to some degree some of the biology that's been seen. We'll take a look in an in vivo model for evaluation and then summarize with an overview of clinical utility and a summary. So with that in mind then, I'd just like to start off with giving an overview of kinases in drug discovery. The kinase world really was set alight back in 1978 with the identification of SRC as the first oncogenic protein kinase. And for those not familiar with the field, by that I mean a kinase that has been linked to cancer. Since then, there have been numerous discoveries along the line. In 1986, Sorafenib was identified as a kinase inhibitor, initially as a fairly potent inhibitor of the protein kinase C family of enzymes. But particularly, Sorafenib is actually a fairly broad spectrum inhibitor and actually has quite a diverse profile. Consequently, this leads to safety issues. So it's not a drug in of itself. And this led to some conjecture in the kinase field initially when it was decided that perhaps kinases might make good drugs. There was a concern that targeting the ATP binding site of kinases would not allow sufficient selectivity such that a safe and selective drug could be identified. perhaps understandable, but as we've come to see in more recent years, and in particular there in 2001, the approval of imatinib for the treatment of leukemias, which is now marketed as Gleevec, really showed the way in terms of being able to identify and develop specific and selective kinase inhibitors that are safe and well tolerated and fulfill their need in modulating human disease. So with that, subsequent to then, there have been numerous other kinase drug discovery programs, and they have resulted in the development of a large number of drugs for a variety of different therapeutic indications. And this is shown on the next slide here. This is an overview of all of the approved kinase drugs since imatinib near the top there. I should point out very quickly that, in fact, it wasn't technically the first kinase drug. Sirolimus, developed by Wyeth, is an inhibitor of the mTOR kinase, but it's a little bit different because it binds to a protein that is essential for the activity of mTOR, and it's not an ATP-competitive drug. But with imatinib and going on from there, there's been a wide range of different kinases that have been targeted, as I mentioned, for several different therapeutic indications. What you'll notice about this is that there are a large number of different kinase inhibitors that are either selective. We have examples there with gefitinib and erlotinib early on in the story, and more recently with midostaurin, neratinib, and ribociclib, which are much more selective than some of the other kinase inhibitors that have been developed. While selectivity can be an important component of a kinase's profile, it is not always an absolute requisite, and in some cases, having multiple activities can be beneficial. Again, this depends upon the therapeutic indication for which the kinase inhibitor is being developed. And as you can clearly see, there are numerous examples of kinase inhibitors that have actually quite broad profiles. Now, what I should add is while they are broad, and in many cases, multiple aspects of that profile contribute to the therapeutic utility of those kinase inhibitors, it should be stressed that they are not so broad that they inhibit other kinases that result in toxic side effects. What I will draw your attention to is sorafenib, which is one of the earlier kinase inhibitors that was approved. And as you can see, while it is not absolutely selective, it has a somewhat broad profile, and we'll delve into some of the implications of that as we go throughout the webinar. I'd mentioned about the pursuit of kinases as drug targets, and so we're just going to overview very briefly here the process of pharmaceutical drug discovery. Starting with initial target validation, and that can take various guises from academic discoveries in a wide variety of different models and practices. Once a target has been linked with a disease, it is then considered a potentially attractive target against which to develop a drug. One typically engages in a high-throughput screen, or the HTS there, shown in the light blue. And I often jokingly refer to this as looking for a needle in a haystack. Sometimes high-throughput screens can involve many, many hundreds of thousands of compounds, if not millions of compounds. And the ultimate goal is to identify suitable inhibitors in many cases, not always. In some cases, the drug target may need to be modulated positively as well as inhibited. But in most cases with kinases, we are looking and generally talking about inhibitors. Once a lead is found, then there is the process of optimization of that lead by dialing in appropriate properties to make that initial inhibitor more like a drug, ultimately to the point that it is safe enough to take into a human being in clinical trials. What I just have here are some of the various parameters that need to be satisfied in order to take a drug forward. And so it is no longer a tool compound, but something that looks more like a drug. And that includes addressing activity. There must be activity at the target of interest. The drug must be potent enough to manifest the effect for which it is being designed. There's no use tickling the target if a more robust inhibition is needed. The drug should be selective. And again, selectivity can be somewhat subjective and depends upon the therapeutic indication for which the inhibitor is being developed. But appropriate selectivity must be achieved. And that ties in with safety. In some cases, if a drug is not selective enough, either within, in this case, the kinase area of targets, but also outside of kinases, that drug may have safety liabilities. The drug must have efficacy, and whereas one will have profiled a drug in cell-based models in the more complex milieu of that, one is also looking at the effect of a kinase drug in an in vivo model, a much more complicated system than a test tube or an assay plate can ever recapitulate with a purified enzyme. In terms of developability, a drug must have appropriate bioavailability, and this is a complexity of multiple different factors, including drug metabolism, the distribution of the inhibitor, and making sure that not only does the inhibitor get the appropriate target, but also the appropriate tissue type in which the target is resident and exists long enough to be able to do the job for which it was designed. As I'd also mentioned, the drug must be safe, and that is perhaps the most important component of this for taking an inhibitor forward into clinical trials. And all of these parameters must be satisfied for a drug to receive investigational new drug status, which is the gatepost that allows going on into clinical trials, the phase 1, 2, and 3 trials there, which we won't be addressing in this particular webinar, but might provide the subject matter for a subsequent webinar. So with that in mind then, looking at a particular case study, we're going to look at the area around BRAF. Now, 15 years ago, a seminal paper came out in Nature that identified the association of BRAF mutation with a variety of human cancers. This was a seminal paper in the field, in that it provided a robust validation of a target, or at least the correlation of a mutation of a target with a human disease. So much so that while melanoma was the first cancer type that was identified, there are others. What I should mention is initially the mutation was identified at position 599. It's a valine to glutamate mutation, but subsequent refinement of the sequencing has now identified that mutation as the position 600, so valine to glutamate at position 600 within the BRAF gene. As I'd mentioned, numerous tumor types have been identified that bear the BRAF mutation, and these are activating mutations that result in constitutive kinase activity, and in many cases give rise to a cancerous phenotype. I'd mentioned melanoma as being perhaps the best well-known there associated with BRAF, but there are numerous other cancers. Some of them are relatively small percentages by incidence rate within each cancer type. But in many cases, for example, non-small cell lung cancer, the population of patients that represent the non-small cell lung cancer group is so large that even that 3% represents a huge number of patients for which medical needs need to be addressed. What I will mention just very briefly is that although BRAF is a component of the mitogen signaling cascade, the traditional RAF-MEK-ERK cascade, it should be noted that the BRAF mutations are very rarely, and in fact less than 1%, found in the same tumors as activating RAS mutations. I mention that because RAS is upstream putatively of the RAF kinases, and it's an interesting correlation or inverse correlation, that the two do not generally tend to exist in the same tumor type. So with the therapeutic rationale set forward, a number of companies set out to identify inhibitors of BRAF as potential therapeutic interventions for melanoma, amongst other tumor types. One of the trailblazers up front with this was Onyx Pharmaceuticals, which identified a compound that was subsequently taken on by Bayer for development and given the number BAY439006. Subsequently, it has been known as Sorafenib, and the structure is shown here. It is traditionally given as a tosylate salt, as many kinase inhibitors are often given in optimized salt formulation purposes. When taking a look at the biological properties of Sorafenib, it is an ATP-competitive multi-kinase inhibitor. We saw on one of the earlier slides that it has several activities against different kinases. It inhibits a variety of cellular processes, and we'll see a little bit about that as we go through the talk. It does have, therapeutically, a broad spectrum dose-dependent anti-tumor activity, and we as a field have seen that in a variety of different xenograft models, and it acts by inducing autophagy and apoptosis in the tumors. So it has biological activity that is desirable for an anti-cancer therapeutic. From a drug metabolism and pharmacokinetics perspective, it has a bioavailability around about 38 to 49%. It is fairly highly protein bound at 99.5%, but it does have a fairly decent half-life, anywhere between 25 and 48 hours. Sorafenib is cleared by the liver, as it is the substrate for the cytochrome P450 enzyme 3A4, as well as UGT1A9. So that's a little bit about the background of sorafenib. So let's look a little bit closer at some of its profiles in some of the assays that we have to offer. One thing I should address before going into that is the importance of selectivity profiling and the information that can be gathered from profiling. Obviously, one can gain information about selectivity, which can have a number of different ramifications. This might include broadening the utility of a particular inhibitor. Often, as we've seen, inhibitors can have multiple activities, and in some cases they can collectively contribute to the activity of that inhibitor, in particular kinase inhibitors as shown here. And in some cases there are targets that are inhibited that lead to safety liabilities. So it is very important to learn something of the selectivity of an inhibitor, because through the power of medicinal chemistry, the appropriate activities can either be dialed in or dialed out as need the compound. One can also predict off-target effects and safety issues around this. For example, a full-scale kinase panel, and we currently at Eurofins offer over 400 different kinase targets that provide a robust platform against which to look. One can take a look and see if there are kinase targets that we know or believe can result in off-target safety issues. And again, that is something that can be addressed during the lead optimization stage. We can also gain insights into downstream effects and cellular phenotypes, and we'll take a look at a couple of the biological studies that get to that, based in the context of the in vitro biochemical kinase selectivity profile. Knowing a little bit more about a broader profile outside the immediate target can also assist in repurposing. And what that means is where a drug might have actually other utilities that can then extend the lifetime of that drug and provide benefit to other patients, sometimes in similar diseases, for example, in other cancers, or sometimes in completely different therapeutic areas. An important thing is to update the profile, and it provides valuable information about a particular inhibitor. Additional targets may also reveal further hits there, and this can, as I'd mentioned before, highlight safety issues. The earlier that is done in the drug discovery process, the quicker that can be addressed, and again, safety issues dialed out at an earlier stage, which ultimately can help accelerate the drug discovery process. One can also feed additional information on leads into ongoing activities, and for example, that can give highlights and information around cellular mechanism of action studies. Collectively, this provides more information about how the inhibitor works, which ultimately enhances our understanding of the inhibitor and can allow more informed decisions about the tumor types or the applications in which that inhibitor is to be used. So the next slide here is showing the sorafenib profile in Eurofins kinase profiler. This is a broad panel of kinases. I'd outlined we have more than 400 kinases now available. And what you'll see here is that sorafenib has a somewhat broad profile against a number of different kinases. Interestingly, and in line with the initial therapeutic direction of sorafenib, this kinase inhibitor is capable of inhibiting all of the RAF family members, CRAF, BRAF, both the wild type and the V600E mutant, and ARAF. So this fulfills the initial goal to which sorafenib was being directed. But what is interesting about looking at some of the other kinases targeted by sorafenib is there are several others of interest here. And I will draw your attention to the examples FLIP3, FLIP1, KDR, which is also known as VEGFR2, or the vascular endothelial growth factor receptor 2, as well as PDGFR beta, the platelet-derived growth factor receptor beta. And the reason I bring those to attention is that they are all targets that are germane to the process of angiogenesis. And that is something for which certain tumor types show a critical dependence, perhaps in some cases more than others. When one compares the profile of Sorafenib with another RAF inhibitor, this is Vemurafenib, that was developed initially by Plexicon and then subsequently taken on by Roche, it shows a slightly more selective profile, and you can see that here in the green box at the left-hand side. Like Sorafenib, Vemurafenib also targets the RAF family of protein kinases and shows significant activity against BRAF, but it targets a few other different kinases, but is generally a little bit more selective than Sorafenib. When one then goes forward and looks in cellular assays, and I'm not going to cover cellular mechanistic assays here, it's important to conduct cellular mechanistic assays because they provide insight into the activity of an inhibitor within the cellular context. They show that a compound has got across the cell membrane and is capable of functioning in a mechanistic fashion within the complex milieu of the cell. But ultimately, the goal of all of these inhibitors is to modulate human disease, and that is manifested by a phenotypic or functional effect. In the case of cancer, we can address this using the OncoPanel cell-based profiling service, which encompasses more than 300 different, diverse, genomically characterized human cell lines, human tumor cell lines, should I say. And those span a wide variety of different tumor and tissue types, 19 in total here. So collectively, one can then look across a wide diversity of different tumor types to gain some appreciation as to the selectivity and the utility profile of a kinase inhibitor, or for that matter, any cancer drug. We perform these assays using high-content analysis. If you look at the bottom left there, this is a typical high-content picture that we see. We're able to evaluate the proliferation, the effect of a drug on cell proliferation by staining nuclei with a DAPI nuclear stain. That's shown by the blue spots, which represent cell nuclei. We're able to also, by using the high content of the feature of this analysis, stain for the induction of apoptosis. We do that by staining for activated caspase 3 as a hallmark of induction of apoptosis. We're also able to evaluate the effect on mitotic status of a cell by staining for phosphohistone H3, and that's shown in the pinkish red spots. One can translate that into dose responses, as seen in the graph above the image at the bottom left, and that shows the typical sigmoidal dose response in proliferation in dark blue, where with increasing concentration of drug, the effect on proliferation is inhibitory, and in this case comes down to nearly zero, giving an indication that the drug is more than likely a cytotoxic drug. In this particular case, I should stress this is not Sorafenib in this particular example, but one sees concurrent with a decrease in proliferation and induction of apoptosis, shown in the green sigmoidal graph, and also an effect on cell cycle in the decrease in the phosphohistone H3 signal there in the pinkish purple. Because we have multiple cell types, we can actually evaluate these parameters across all of the panel, and we can gain an appreciation for which tumor types are more sensitive or less sensitive to any particular drug, and that can be measured by a variety of different cell proliferation parameters shown by EC50, IC50, GI50, and they all give very slightly different information about the activity of that drug, and I'm not going to go into detail with that here. We addressed that in the last webinar. But as I mentioned also, we can look at the induction of apoptosis or the change in phosphohistone H3 signal as a result of affecting the cell cycle, and that might either be an increase in the signal or a decrease in the signal. So this is the platform that we can then profile oncology drugs against. And this is an example for Sorafenib. So when we look at Sorafenib in the OncoPanel profile, we see the following. There is activity against a certain number of cell lines, and they're showing activity here at a single concentration. So on the far left-hand side there are the cell lines that are most sensitive. We're plotting the log of the IC50 in nanomolar, and each one of the vertical bars represents an individual cell line. So you can see activity here in the green box. But what is interesting is, given that Sorafenib is a BRAF inhibitor, amongst other things, is there does not seem to be a robust correlation between the presence of BRAF mutations in tumor cell lines and the activity of this compound as far as inhibiting proliferation of those cell lines goes. I've also included the indication here of cell lines that either have activating mutations in NRAS, and we have 2 or 3 of those in black, as well as overexpression of c-KIT, and those are shown in yellow. The reason I mention these is because putatively they are upstream of BRAF and can potentially activate BRAF, and thus if you're inhibiting BRAF as a downstream target, it might be plausible to expect that a BRAF inhibitor could inhibit the dependence of those cell lines on either activated NRAS or c-KIT, overexpressed c-KIT, resulting in a decrease in proliferation. But again, we don't see that here for Sorafenib. If one compares again with Vemurafenib, you see a slightly different profile. What is interesting here is this has a somewhat broader profile, at least within the context of the cell lines that we are examining with an OncoPanel. But when you look at the correlation, you see the most sensitive cell lines there cluster very tightly with the presence of BRAF mutation. Each one of the bars with a red coloration is a cell line that has a mutation in BRAF. This is such a strong correlation that we actually see an associated p-value of 10 to the minus 22, highly significant. And this is borne out by the use of Vemurafenib in the clinic. Ultimately, Vemurafenib has become the marketed drug Zelboraf and has been approved for the treatment of metastatic melanoma bearing BRAF activating mutations, amongst other tumor types. So when one looks then at the profile of the drug, and one looks carefully at the genetic composition of the cells, of the cell types being affected, one can begin to appreciate that there may be genomic characteristics that correlate with sensitivity, and by corollary, correlate with resistance. What I should mention here at this point is that kinase profiler and OncoPanel are complementary approaches, and it's important to take the activity of both together. No one piece of information in any drug discovery program should be taken in isolation, and the collective pool of information should be taken together to give a fuller picture of the biology and the activity of a compound, ultimately to enhance our understanding of its ability to modulate disease. Being able to translate in vitro enzyme activity into a cellular context is a key step in that. But not only doing that, providing a broader understanding of the biological profile can help provide ultimately a broader understanding of the potential clinical application of that compound. This can collectively, again, comparing the 2 different formats, can gain deeper insights into the biological consequences of inhibition. And in some cases, and we'll take a look at some of the biomarkers, that may also lead to the identification of biomarkers that could lead to resistance mechanisms, again, providing avenues of approach to address that clinically and ultimately to provide greater benefit for patients in need. Being able to identify new identify and evaluate new properties for asset repurposing as another important point of being able to evaluate these 2 different approaches together. And just lastly, at the bottom there, gaining a fuller picture up front allows more insightful in vivo model choice. There are a lot of initiatives out at the moment that are aimed at reducing and refining the models that are used for in vivo research. It is an unfortunate necessity of drug discovery to be able to test a compound, a would-be drug, in an in vivo model. But from the humane perspective, one is always looking to try and reduce the numbers of subjects needed in those studies, while still providing valuable information in being able to develop and progress that drug. One of the important things here for Sorafenib, as we'll see, is the ability, using these pieces of information, to identify and evaluate new properties for asset repurposing. And we'll see a little bit about how that plays out with Sorafenib as we go forward. What I would like to go into very quickly here is how we identify clinical biomarkers, potential clinical biomarkers, at least biomarkers that are predictive and can help streamline oncology clinical trials. I'm sure many of the audience have seen the Hallmarks of Cancer, the seminal paper from Hanahan and Weinberg, that outlined many of the genetic aberrations that drive cells to become cancerous. And traditionally, when one looks at the structuring of clinical trials, in the earlier days, the shotgun approach was taken, whereby the trials were very large and very costly. And because no stratification based on genetic biomarkers or anything like that was taken into account, unfortunately, the probability of success of those clinical trials was relatively low. And ultimately, the degree of benefit for the patients enrolled in those trials was also similarly low. Now, with the ability to identify predictive biomarkers and thus stratify the patient population into which the drug is being taken for clinical testing, this has numerous benefits. First off, the trials can be made smaller and more refined. This results in a lower cost for the clinical trials, but it also results in a higher probability of success, which ultimately benefits the patients. Being able to identify a certain mutation or overexpression of a gene that gives rise to the cancer of interest, that allows the clinical trial to be more streamlined. And we see that with the examples here, whereby aberrations in the genomic components of a variety of different tumors were used in the streamlining of trials that gave rise to these drugs. A very successful approach, and something that can and is being used going forward. Just coming back briefly to the overview of the OncoPanel profiling service that we talked about earlier, because we have a large number of genomically characterized human tumor cell lines there, we can evaluate the performance of a drug against those cell lines and tease out statistically which genetic components of each tumor cell line that is either sensitive or resistant to that drug contribute either to that sensitivity or resistance. With that in mind then, this is an example for Vemurafenib, whereby if we look at the profile of Vemurafenib across that collection of tumor cell lines, we can identify which particular genomic markers are significant. We do that by evaluating the drug response in two different statistical tests. Every statistical test has an inherent bias of some form or another, and we feel that doing two statistical tests orthogonally helps to reduce that bias, so much so that if one employs the Fisher's exact test and the student's t-test, we can identify genomic sequences here that we deem to be more significant than the bias provided by one test alone. By evaluating those markers further, we can actually look at the odds ratio versus the effect, but we can actually see which of those biomarkers correlate with resistance or sensitivity. And perhaps for Vemurafenib, this is not entirely surprising. Visually, we saw from the OncoPanel profile that there was a very strong correlation between the presence of the BRAF mutation and sensitivity. This is borne out here both statistically and within the context of predictive biomarkers. You will see in the right-hand splatter plot, in the bottom left quadrant, the sensitivity quadrant, the strong presence of BRAF mutation or BRAF mutation associated with amplification of that pathway. As I mentioned earlier, the ability to identify predictive biomarkers is valuable not only in being able to streamline clinical trials based on markers for potential sensitivity, it can also identify markers for potential resistance. And we see that here with examples for the BRAF pathway as targeted by Vemurafenib. It's perhaps not surprising that cell lines with mutant KRAS, where KRAS has many effectors, can actually signal through other pathways, wherein BRAF inhibition alone does not provide enough of an inhibition to dampen or inhibit the proliferation of those particular tumor cell lines. Again, there are other markers that can be correlated with resistance. And when I say resistance, that is either insensitivity or true resistance. So going back to our case study then, we're looking at Sorafenib here. This is an example of the waterfall plot. We've seen these on previous slides where I colored each of the cell lines with a different color. This is just simplified now into blue going through to red as one goes from the less sensitive, or for want of a better term, resistant cell lines through the spectrum of activity to those that are more sensitive. It's important to set cutoff limits for the statistical evaluation because in doing so, one can then focus on the true genomic components that make up the most sensitive or the most resistant of the cell lines that are being evaluated. If one extends those cutoff values towards the middle of this particular chart, the ability to pull out statistically meaningful samples is diminished. So moving forward then as to how the plots look for Sorafenib, this is the initial evaluation where we're pulling out markers here. So we only pull out 2 markers with Sorafenib, and this is based around mutation. So when one looks at the sensitivity versus resistance plot, we see the 2 markers correlate with sensitivity. And interestingly enough, neither one of them is BRAF or associated directly with the BRAF pathway. In fact, interestingly, in both of these cases, they're related to gene transcription and protein translation, chromatin organization. So a very different component than the signal transduction markers identified with Vemurafenib. One can also, with the statistical analyses, look at which genes are differentially expressed and how that correlates with resistance or sensitivity. And that is summarized in this table here. And I'm not going to go into great detail here, but we can identify the differential expression of which genes correlates either with sensitivity or resistance. And we see a number of different genetic signatures here. Again, some of them, again, correlating with ribosomal processes, with the general process of gene transcription and going forward into protein translation. In addition to that, with respect to resistance, we see there towards the bottom SMAD3 is being identified. This is a key player in the transforming growth factor beta signaling pathway. One can make rational cases for why that might provide an advantage against an anti-cancer drug. So there's a lot of information that can be gathered from a genomic analysis such as this. But when we want to look at the true effect of a drug in a much more complicated scenario, we have to go to an in vivo model. And this is an example of some of the portfolio of assays that can be conducted by our partner laboratory, the Pharmacology Discovery Services Unit in Taipei, Taiwan. They have a wide range of experience across a broad range of therapeutic areas and have numerous models of human diseases. For the sake of this case study, I'm going to focus solely on the oncology area towards the bottom there. And I'm actually going to show you a syngeneic model, which is a mouse model that has a fully functional immune system, wherein we can take a look at how Sorafenib performs and how it compares with a couple of other comparator drugs. So looking at that then, this is an example of one of the syngeneic models that Pharmacology Discovery Services offers. This is the Renker kidney cancer model. And what I should cover first is the black line shows the no treatment, typical progression of the tumor volume. Without treatment, the tumors grow. Upon treatment with a variety of immunotherapeutic agents here, these are immune checkpoint blockade inhibitors, either targeting CTLA-4, PD-L1, or PD-1, and these have become targets that have been very much in vogue in the oncology drug discovery environment recently. This particular xenograft model shows that while they don't have spectacular activity, perhaps the anti-PD-L1 agent here actually has a reasonable activity. What is striking is that Sorafenib actually has great activity in this model. I say this is striking because the tumors are being put essentially on stasis, or the growth rate of those tumors is very significantly diminished compared with the no treatment control. And what is significant here is that Renker as an example of a kidney cancer, ultimately correlates very well with the clinical utility of sorafenib, and we'll discuss that in the slide coming up just now. So sorafenib went through a number of clinical trials. Initially, as I mentioned, it was put forward with the idea of targeting BRAF mutations in melanoma. Unfortunately, it did not show the therapeutic utility that was hoped for. But interestingly, there is a subset of melanoma patients that did respond very strongly to sorafenib, so it did have some benefit in that patient population. However, and it's very nice to see that it correlates with the in vitro studies that we've just discussed, none of those patients that benefited had the BRAF mutation. And in fact, the authors of the papers outlined at the bottom make the case that there is the need for more biomarker identification here to help understand how some of these unknown clinical effects are manifested. And again, it comes back to the point of understanding as much of the biology and the profile of a drug as one can, and being able to translate that into observed clinical effects or to be able to predict clinical effects. I'd mentioned the Renker model that we have, that we identified, that we evaluated sorafenib in. Ultimately, sorafenib, when it was put into kidney cancer trials, showed great effect in the clear cell renal cell carcinoma. It showed an increase in progression-free survival of nearly 3 months against placebo, and this ultimately led to its approval in 2005. Hot on the heels of that was the identification that sorafenib had utility in liver cancer with similar improvements in survival, and that was then approved in 2007. Interestingly, sorafenib was ultimately approved, as I mentioned, for the two cancers, the renal cell carcinoma and hepatocellular carcinoma, and it's been approved as the drug Nexavar. But it has also been approved for I-131 resistant thyroid carcinomas. And I didn't mention this in going through the profile, but from the oncopanel profile, when one looks at some of the tumor types that are sensitive to sorafenib, we do actually see a couple of thyroid tumor cell lines that are sensitive. So again, this agrees retrospectively with what was seen in the clinic. So although sorafenib didn't fare so well in the initial indication for which it was put forward, it has nonetheless shown itself to be a very valuable drug in the oncology therapeutic area, and has shown great effect for patients in need that are both suffering from renal cell carcinoma and hepatocellular carcinoma. And further investigations have been ongoing to see other utilities for sorafenib. So to wrap up then, I'd just like to summarize some of the learning points from today. We've shown that evaluation of a drug's kinase profile can provide a wide range of different indicators, not only from the potential for dialing in a safer profile, but also potentially from providing indicators of potential utility ultimately in the clinic. We saw for sorafenib that while it does show appreciable activity against RAF kinases, it also shows activity against a number of anti-angiogenesis targets that lead to anti-angiogenesis. And that ultimately can go some way to explain its clinical utility. Both renal cell carcinoma and hepatocellular carcinomas are more heavily vascularized tumors. Cell-based profiling can also help to validate and in some cases refute aspects of the biochemical profile. We saw that again with the case study here with sorafenib, especially when one compares it with other agents, as we did here with vemurafenib. Genomic analysis, as we've seen, can provide a very powerful tool for correlating phenotypic response or drug response with identifying potentially meaningful predictive biomarkers, not only for sensitivity, but also of potential resistance. And that ultimately can help improve the design of the molecule as it goes through lead optimization, and ultimately, once a drug has been put into clinical trials, can begin to provide ideas as to combating potential mechanisms of resistance. I also mentioned there that this can also enable more logical selection of in vivo models. I'd mentioned previously that there are a number of initiatives around the world to reduce the number of in vivo models that are used, but we still need to be able to identify models that provide meaningful information that allow the progression of drugs. Ultimately, they have better therapeutic effects and safer profiles when put into human beings. So collectively, what I'd like you to take away from this webinar is that no one piece of data should be taken in isolation. And it's actually the complete profile and making sense of the observations of all of the different aspects of the evaluation that allow us to more reliably paint a picture of potential clinical utility. So with that, then I'd just like to quickly give you an overview of some of the things that we can do at Eurofins Pharma Discovery Services. I'm not going to go into great detail here, but we provide essentially a full-service portfolio of assays, including now medicinal chemistry, that we can help all aspects of the drug discovery process. And we can do that especially with the inclusion of in vivo models with our partner lab at Pharmacology Discovery Services. So with that, I'd like to thank you all very much for your time. I hope you've all learned something today. And if you have any questions or would like to follow up with us, please visit us at the website shown here or drop us a line at the email address shown. So with that, I'd like to thank you all once again.
Thank you very much, Dr. King, for your presentation. And I'd also like to thank Eurofins Pharma Discovery Services for their sponsorship of today's web symposium. I would like to thank you on behalf of Cambridge Health Tech Institute Global Web Symposia Series and the folks at Eurofins. Thank you so much for spending this time with us, and have a great day.